Okay, then I will start, and if I think I need to stop and have a break, then I will tell you. There's a small fly here. I didn't get mad in the meantime. Okay, let's start. Today I'd like to show you how EULAR recommendations are developed, and I will use the example of the 2022 update of the ASAS EULAR recommendations for the management of axSpa. I'm Desiree van der Heijde. I'm Professor of Rheumatology at the Leiden University Medical Center in the Netherlands. These are my disclosures. And we use for the EULAR recommendations the standardized operating procedures that have been published in 2014. Currently, there's an update ongoing, so in the future, I don't know exactly when, you will find new updated standardized operating procedures for these EULAR recommendations. First, you start with a project proposal. You define the project and a task force, and that's done by a convener and a methodologist. The convener is the person who has the idea and wants to run the project. A methodologist is a person who knows about the process, how it should be done, and this person should be endorsed by EULAR. So if you have an idea, then first look at the EULAR website for the approved list of methodologists. Then you submit that proposal to the chair of the EULAR Quality of Care Committee. That person has all the recommendations in his portfolio. There will be an evaluation of the EULAR board, and assume that it is accepted, then you can start the process. Then the convener has to invite the task force members. There are certain rules. So you need to have a sufficient number of countries which are represented. Also, IMUNET members, patients need to be part of the task force, healthcare professionals other than rheumatologists. So there are certain rules that you have to follow in inviting task force members. Then usually when it's the first time for recommendations, there will be 2 meetings. And usually when there's an update, there is frequently only 1 meeting. So if you start with new recommendations, you have the first meeting of the task force, and then you define which questions need to be addressed by systematic literature review. Thereafter, that SLR is performed by 1 or more fellows and supervised by the methodologist and the convener. Thereafter is the second meeting of the task force, and then the most important part is a presentation of the results of this SLR, and thereafter the formulation of the recommendations. Then that steering group with the convener, methodologist, and fellows, they give the level of agreement and the recommendations. First, the grade of recommendations, and thereafter all the task force members have to assess the level of agreement with the recommendations. Then the abstract that will be drafted has to be presented first at EULAR. You cannot first present it at another conference, and that needs to be approved by the chair of the Quality of Care Committee. Later, you submit the manuscript to ARD, but only after approval by the chair of the Quality of Care and the board of EULAR. Thereafter, you could consider if you want to develop also a lay version. Sometimes that's also done by ARD itself. And then important is the dissemination and the implementation. I will give you some more background of all these different steps. So here was the steering committee of the ASAS EULAR recommendations for the update of axSpa. You see there are 2 methodologists here. One is the official approved, and another one is a junior methodologist. And if you want to become a methodologist approved by EULAR, you first have to follow, I think, 2 task forces as a junior methodologist, and thereafter you can be a senior methodologist. The fellows, we had 2 in our project, are extremely important, and they do really a lot of work, which is the basis for the management recommendations. Then you have a task force. This was a task force for the axSpa recommendations. You see we had a wide representation of different countries, 16 in total, and we had different healthcare professionals, rheumatologists, epidemiologists, healthcare professionals such as physiotherapists, PARE, that's the representation of patients, and IMUNET, which are the young EULAR members, and young ASAS, which are the young ASAS members. And then in the process, you also have members that are proposed by national societies. So when there's a new task force for recommendations that's published on the EULAR website, and then as an individual, you can apply to your national society, and the national society can then also propose you, or you can apply yourself. Then this is a combined project with ASAS. This is an exception. Usually it's just EULAR projects, but this has a long history, and that's why it's still a combined project. So important are the 2 SLRs. We performed 1 SLR on the efficacy and safety of non-pharmacological and non-biological interventions. So in fact, these are the CS-DMARDs and TS-DMARDs, and also another one on the efficacy and safety of biological DMARDs. We had, this was done by the steering group, and then we had a 1-day task force online meeting because of the COVID situations. And there we discussed the overarching principles and the update of the recommendations. And I will explain later how we did that exactly. First, you have to define the target users. So for whom are you developing these recommendations? In this case, it's all healthcare professionals taking care of patients with axSpa, also patients with axSpa, but also other stakeholders. So this is really a very broad group of target users. If we then go to the process of the SLRs, you start with PICOs and the search strategy. Then we did a registration in Prospero. That's an important step. We did the search, the output, the data extraction, the risk of bias, and then you have the final review and the presentation of the slides, first in the steering group, and then later for the entire task force. For EULAR recommendations, we use the Oxford level of evidence and grade of recommendation, and that's presented here. So for each study, you first have a level of evidence that goes from 1A to 5. 5 is the lowest. That's only expert opinion, while 1A is a systematic review of various RCTs of homogeneous, high-quality RCTs. And then a combination of the studies that is available that provides the grade of recommendation. In EULAR, we are not using the grade methodology. This is completely different. There you have different small research questions, and then you also get many different answers, and that brings you to a very high number of recommendations with a high level of evidence or a low level of evidence, and that's very detailed and very difficult to understand for practicing clinicians. That's why user is still using this methodology. Here's an example of the PICO that we use for the efficacy SLR. This was for the biological DMARDs. So we use all types of biological DMARDs and we compare them to everything that's possible, and we defined the outcomes that we considered important. There's a different PICO for safety, again, the example of the biological DMARDs, and also, of course, you have different outcomes. What is important that as this was an update, you only search the literature since the previous SLR. There's no language restriction. And for the efficacy, we used RCTs, controlled clinical trials, open label extensions, and long-term extensions, all for efficacy, and the latter also for safety. And we have cohort studies and registries which we use for safety. We can use meta-analysis of SLRs, but only if they are performed by Cochrane. And we use qualitative studies from the patient perspective. Then we look for full text articles and conference abstracts, but only from EULAR and ACR and from the last 2 years. We expect that if you have an important study, that will be published within 2 years after publishing the abstract. Then what is really important is the risk of bias assessment. And for RCTs, we use the Cochrane risk of bias tool, version number 2. And for observational studies, the Heijden's quality in progress studies, the GRIPS tool. So these are those risk of bias tool. In the end, you get either a low risk of bias, so that's what you want to have. Unclear, that's usually if you do not have sufficient information, or a high risk of bias tool. And abstracts, you do not have the information, so this is usually not available for the risk of bias study. So this is the example of the Cochrane tool, and this is the example of the GRIPS. I will not discuss in detail, but you can find them in the literature. After we have performed the SLR, there need to be a synthesis to be able to present it to the entire task force. And first, we have a summary of the previous SLR so that we have all that information available, and then we add what's new. We give the data as detailed as possible. This is an example of one of the tables. This was just on one biological DMARD, one comparison, one IL-17 inhibitor in this case, where the most important is the risk of bias column at the end, to the right. You have the L for low and the green coloring for low, the yellow for unclear, and the gray for no info. So you can see immediately which studies are important, which provide relevant and qualitative high information. And then important is the primary endpoint. If it's in green, then it was met. If it's in red, it was not met. Then another example is then how you summarize it. This is just a slide on what's new for the entire class of IL-17 inhibitor, and also here the information if it was based on a study with low risk of bias, unknown risk of bias, of unclear risk of bias. And you get similar data for the safety. So also this was again presented first the previous SLR, the current update, and this was done by drug class. So also this is the example of safety, what was new. This is the example for TNF inhibitors, and again you see a global overview of all the studies that we found in the SLR. This is an example of the table of the non-pharma research. You see immediately that this is much more complicated. Also, the risk of bias is completely different. Here you most frequently have a high risk of bias. If you are lucky, it's unclear, but it's very infrequent that you have a low risk of bias. Also, the way you analyze these data is different. It's usually with with effect sizes, which is also more difficult for interpretation. After we show all the data from the SLRs, there's a discussion within the task force. They can ask for clarification, and then we draft the updated recommendations. Usually there's a proposal of a draft recommendation done by the steering group. Then this is discussed by all the task force members. And then we start discussing wordsmithing. Should it be different, a few words extra here, or is the meaning okay, but we should word it different, or do we not agree with the meaning? And then this is working on the wording. And thereafter is an official vote. So we have a round 1 of voting, and then to accept that recommendation, there needs to be agreement by at least 75% of the task force members. If we do not agree, if that's less than 75%, then there'll be a new discussion, and there may be some changes in the wording. People say, yes, but I would agree if I would make a change of this. And then we have a second round of voting, and then there needs to be a 2/3 acceptance. And if that's not the case, then again, there will be some discussion and wordsmithing, and then round 3 is just simple majority. For an update of the recommendations, it's really important that you only change an overarching principle or a recommendation if there is new evidence. So not just because you think the wording was not so nice last time and I want to have a different wording now. No, only if there's new evidence. And also, once a recommendation is agreed and voted upon, it should not be changed. So it cannot be that we have a recommendation, we agreed in the voting, and then 10 recommendations later someone says, I want to go back to recommendation 2, I want to make a change. That's not possible. After the meeting, there is by the small steering group and especially the fellows and methodologists, there's addition of the level of evidence for each recommendation and the grade of recommendation. Usually that's for can be for a small part of each recommendation, because if you have several aspects within one recommendation, they do not all have the same level of evidence. And with this level of evidence and grade of recommendations, all recommendations in one table are sent to the task force members. And for each recommendation, they have to give a score. I do not agree at all, from 0 to I fully agree, 10 on a numerical rating scale. And this is then presented as a mean, the standard deviation, and also the percentage of task force members who gives a score of 8 or more. So this is an example of the overarching principles. That was the end result of the update. And you see the level of agreement, and also the percentage of task force members that score an agreement of a score of 8 or more. And here you do not have the level of evidence, because that doesn't apply to overarching principles. So overarching principles are those things that are so obvious that you always have to do this. For example, that you discuss everything with the patient, and how severe the disease is, etc. Then you have the recommendations, and for this update we had 15 recommendations. 8 are unchanged from the previous recommendations, so there was no new evidence. 3 were minor edits that was mostly on nomenclature. For example, the term extra-articular manifestations has in the meanwhile changed to extra musculoskeletal manifestations, so we made that obvious change in the recommendations. Then we had 2 significantly updated recommendations and 2 completely newly formulated. When we have all those recommendations present, then we try to combine this all in a figure, so that hopefully it will be helpful for someone to get all the information together. This is the figure that was presented at EULAR. Currently, we have an updated figure just with a different layout. The content is the same, and I think that will look much nicer, less crowded than this one. So I hope that you will like the figure that will be published in the end more than this one. This is the general idea. And so in what we had, in fact, in this update is that we addressed the entire spectrum of axSpa, non-radiographic, radiographic, but also the non-pharmacological and the pharmacological treatment. And there is again importance of the non-pharmacological treatment, because we think we want to discuss a lot about pharmacological treatment, but still this is the cornerstone. Also, NSAIDs remain the first-line pharmacological treatment. We stated criteria for the start of biological and targeted synthetic DMARDs. A change was that only ASAS 2.1 or more is selected as a high disease activity criteria, so no longer BASDAI at least 4. Also, the indication for biological and TSD MARD is either a TNF inhibitor, an IL-17 inhibitor, or a JAK inhibitor, but the current practice is to start the TNF inhibitor or an IL-17 inhibitor, and that has to do with the oral surveillance study, where we have the safety study, which revealed a high risk on cardiovascular and MACE side effects in specific risk factor groups. There was a new evidence recommendation on extra musculoskeletal manifestations when you have uveitis or IBD present, and also about when there is treatment failure. This is a new recommendation that this should trigger a re-evaluation of the diagnosis, and also consider the presence of comorbidities which may influence the disease activity level. So you think there's active disease, but it's caused by another comorbidity. And if active axSpa is confirmed, then you have to switch to another biological or TSD MARD. And there's also a recommendation on the tapering of the biological DMARDs if a patient is in sustained remission. So this is in a nutshell how you develop, and in this case update the recommendations according to the EULAR guidelines.
Thank you very much.